Hey guys, today we're moving into the topic of biotechnology. So make sure before you view this lesson that you have read pages 270 to 275 in the Biology in Focus textbook. Again, biotechnology is our focus today, and we'll begin by just defining that term. So biotechnology is any technological application meaning putting knowledge to work, that uses biological systems, living organisms, or anything derived from a living organism, such as DNA or protein, to make or modify products or processes. So that's kind of a mouthful. But basically what it boils down to is using our knowledge of DNA, our knowledge of genes, to solve problems, to improve crops, to synthesize proteins for medical use, to solve crimes. There's forensic applications of biotechnology. And one of the techniques that falls under the umbrella of biotechnology is genetic engineering, which is any direct manipulation of genes for practical purposes. And it usually involves moving genes from one organism to another, often from one species to another. Okay, so let's go through some terminology that is important that you'll need to know to understand this lesson. First, we have recombinant DNA. Notice it has the root word combine. Recombinant DNA is DNA in which genes from two different sources, and that could be two different cells, two different species even, have been combined. So it's a combination of DNA from two different sources. When that recombinant DNA is introduced into a cell or into an organism, that organism now becomes a transgenic organism. And a synonym for that term is genetically modified organism, which I think is the one that maybe we're more familiar with. We've all heard the acronym GMO. Genetically modified organism. It's just an organism that has been, well, genetically modified by artificially adding a gene that would normally not be present in that species. And again, usually that's adding a gene or moving a gene from one species to another. Another term that comes into play here is the term vector. In the context of biotechnology, a vector is a fragment of DNA that functions as a vehicle. That is, it allows a gene or genes to be inserted into a target cell. So a DNA fragment that functions as a vehicle for the insertion of a foreign gene, a gene that would not normally be present in that cell, but we're going to use the vector to move it into the cell. Now, there are lots of different vectors that are used. I've listed 3 here. There certainly are others. We're going to focus on the first one the most in the examples that will follow. And it says recombinant bacterial plasmid DNA. So this is DNA, a small piece of DNA that's taken from a bacterial cell. A gene, an extra gene, a foreign gene is added to that bacterial DNA, and then the bacterial DNA, the plasmid, as it's called, is then reinserted back into a living bacterial cell. So we're using the plasmid as a vector, a vehicle for moving a gene into a cell. Viruses can also be genetically engineered to transport uh, genes, and not only transport them, but actually insert those genes into cells. And probably the most bizarre vector, this is often used to genetically modify plant cells, plant tissues, is what's called the DNA gun. And I've got a picture here. If I can get this to, there we go. So you see this technician is holding this, the kind of the barrel of this DNA gun, and it literally uses gunpowder, as you see here, like a real gun. At least the early ones did. I don't know if they still do it this way. And when the bullet, quote unquote, is fired, out comes these plastic pellets at a super high velocity, and each little pellet is coated with a small amount of DNA. And it's the DNA, it's the gene that you're trying to introduce into this plant tissue. And so it sounds kind of crude, but it is pretty effective at genetically modifying plants. So you can take embryonic tissue and fire the gene gun and introduce genes into those cells. Okay, so I mentioned earlier that one of the types of vectors that is used pretty frequently in biotech labs today is the bacterial plasmid. So here's a picture of a typical bacterial cell, and as you know, they are prokaryotic, so the DNA is not stored inside a membrane enclosed nucleus. It's just kind of naked DNA that's floating in the cytoplasm. And for most bacteria, there's one large chromosome where most of the bacterial genes are located. And then there may be several very tiny loops of DNA that only have a small number of genes. Those little loops of DNA that are separate from the main chromosome are called plasmids. And there are all kinds of natural processes, conjugation and transformation, natural processes by which these plasmids can be shared back and forth between bacterial cells. Sometimes copies of plasmids are made and then shared to other bacteria. So that happens naturally sometimes, but what we're kind of focused on here is, in a laboratory, the ability to extract plasmid DNA from a bacterial cell, and then once you have that plasmid out of the bacteria, then you can manipulate it, you can add genes to it, and then reinsert that plasmid into living bacteria to genetically transform them, to genetically modify them. Before I go any further today, I would like to honor a man who is not only a living legend in our athletic program here at Union Pines, but also happens to know a great deal about bacteria and a great deal about biology in general. Today's code word for extra credit is Coach John Fry. Coach John Fry is our extra credit code word today. All right, so I'd like to walk you through the process of how bacterial plasmids can be genetically modified and how bacteria can then be used to clone a gene of interest. So if we have a gene that we want to make many, many copies of, one way to do that is to introduce that gene into living bacteria using a recombinant plasmid, and then allow the bacteria to go about their normal reproductive process, copying the DNA, making many, 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 many copies of the gene. So I'm going to walk you through that process next. Okay, so the first step is to isolate a plasmid, to get plasmid DNA out of living bacteria, and typically that's done by killing the cells, lysing the the membrane and the wall, and then extracting that plasmid DNA. The second step is to isolate a gene of interest. That simply means some gene that codes for a protein that is important to us in some way. So let's just use as an example the gene for human insulin. All right, so let's say this is a human cell, and this is the DNA from that human cell, and somewhere on that DNA right there is the gene for human insulin. Now, as you recall, insulin is a hormone. It's a protein that's made by humans and other mammals, and it regulates the level of glucose in the blood. And people who suffer from the disease diabetes often need injections of insulin to treat their disease. So for a very long time, medical insulin to treat diabetes was derived from animals. You know, it was rabbit insulin or cow or pig insulin, which works okay in humans, but it doesn't work great. It would be better if we had a cost-effective, efficient way of making real human insulin. And so this was one of the first big success stories of biotechnology, the ability to use GMO bacteria, genetically modified bacteria, to make real human insulin. So to do that, you have to take a human cell, you extract the DNA from that cell. You don't have to kill a human to do that. You don't even have to have a pancreas cell. It could be any somatic cell in the body. And you must cut out just that one gene that you're interested in this case, the human insulin gene. And you're going to introduce that gene of interest into this bacterial plasmid. So you see why we call this recombinant DNA. We're combining bacterial DNA, which is here, with, in this case, human DNA, and we're combining them together on one piece of DNA. We're making a combination of bacterial genes and a human gene, thus we call it recombinant DNA. Now to do this, to add a gene to this plasmid, it's a closed loop, so you must first cut an opening in that plasmid. And I said earlier, you have to make a cut on either side of the gene of interest here to remove just that one gene. So how do you cut DNA? Well, that's a side story that's sort of a story in and of itself. You can't cut DNA with scissors. So how do you cut DNA? Okay, so to cut DNA, one must use an enzyme called a restriction enzyme. Sometimes they're called restriction endonucleases. These are enzymes that are naturally occurring in bacteria. They were first discovered by science in the 1960s. And the reason these enzymes exist is because if a bacteria is infected by a phage, a bacteriophage virus, its only defense against that virus is to destroy the virus DNA. Remember, when a bacteriophage infects bacteria, it does so by injecting its own virus DNA into the bacterial cell. So if you're that bacteria, you have a very short window of opportunity to destroy that viral DNA before it has a chance to reprogram the cell. And so that's what restriction enzymes do. They cut DNA. And they don't just cut it randomly. Each restriction enzyme has a very specific sequence of nucleotides that it recognizes. And these recognition sequences that they're called are always palindromic. They're palindromes. Now, what does that mean? That means if you read it forward, GAATTC, and then you drop down to the complementary strand and read it backwards, you get the same thing, GAATTC. Each restriction enzyme recognizes a different palindromic sequence on the DNA, and it will make a cut. It'll literally cut through the DNA, breaking it into two halves, but it'll only cut when it recognizes that particular recognition sequence. Now, there are hundreds of different, maybe even thousands of different restriction enzymes, but one that's fairly commonly used in biotech labs is called ECHO-R1. And ECHO-R1 does indeed recognize GAATTC as its recognition site. By the way, the names of these restriction enzymes has sort of a formula to it. So the first three letters indicate the species of bacteria where this enzyme was first discovered. So pretty easy to figure out. This was E. coli. And then the R stands for restriction enzyme. And then there's a Roman numeral, and in this case it's a Roman numeral 1. So this is the first restriction enzyme ever discovered in E. coli. Now, the beauty of a restriction enzyme is that when they cut through the DNA, they often create these little single-stranded overhanging pieces called sticky ends. And that's because the cut is not a straight across cut. It's usually kind of a zigzag cut. And these little single-stranded pieces that hang out where the cut was made are very useful. Because if you have a piece of DNA that you've just cut, and then you have a piece of DNA that you got from some other source, some other cell, if you cut this new DNA with the same restriction enzyme that you cut this DNA, the sticky ends will match. See here, the AATT will match up with the TTAA. They'll hydrogen bond, they'll stick together. And down here, the TTAA will hydrogen bond with the AATT. It makes it easy to insert DNA from an outside source and combine it with the DNA that you're trying to modify. So when you see in this picture, when it talks about cutting out the gene of interest or cutting an opening in the plasmid, those cuts are made by restriction enzymes. Okay, now it's a little more complicated than we see in this diagram, because after you insert the gene of interest into this plasmid, and this is now a recombinant plasmid, there are still some little tiny gaps that have to be sealed up to make this a solid piece of DNA again. And that is accomplished by an enzyme that we've talked about before back in when we were learning about DNA replication. It's called DNA ligase, and it forms covalent bonds here and here to seal the gene of interest in place. So now we must get that recombinant plasmid to physically go back into a living bacterial cell. Now, there's a lot of techniques for doing this, but one of the oldest techniques, and the way we would do it in an AP biology lab, is through a combination of cold calcium chloride and heat shock. So what we would do is we would take some living bacteria, put them in a test tube with a cold solution of calcium chloride. And for some reason, I don't think the reasons are completely known, for some reason cold calcium chloride causes very tiny openings to develop in the membrane around the bacterial cell. It causes the membrane to become somewhat porous so that it's physically possible for plasmid DNA to get through those tiny holes and get into the bacteria. Now, the fancy term for this in biotech labs is we're going to make the bacteria become competent. We're going to render the bacterial cells competent to receive the plasmid DNA. Another way you do this is by heat shock. So you take the test tube, which has the bacterial cells and the plasmid DNA all mixed together, and you put the test tube on ice, make it very cold, and then you quickly transfer it into a 37 degrees Celsius water bath, which heats it up. Or actually, I think it's even warmer than that. I think it's like 40, maybe it's 40 or 42 degrees. Anyway, you heat it up, and then you quickly transfer it back to the ice. That very sharp change in temperature causes those little tiny openings in the surface of the membrane to become even larger, which makes it easier for that plasmid DNA to get in. Okay, so if you successfully get bacteria to take in this recombinant plasmid, even if you just get one bacterial cell to take in this plasmid, that is now a recombinant bacterium. It now has its normal bacterial chromosome, it has its normal bacterial genes, but it now also has this gene of interest. In this case, the human insulin gene. Now you might wonder, well, how do you know? How do you know if you've been successful at getting the bacteria to take up the plasmid? Like, how do I know that this bacteria is actually going to be able to make some insulin for me? Well, one way to know that, and this is kind of clever, I think, is in addition to introducing, say, the human insulin gene, on the other side of the plasmid, you could add another gene. And oftentimes, this other gene will be something like an ampicillin resistance gene, a gene that makes bacteria resistant or immune to a particular antibiotic. And then what you do is you take the bacteria and you introduce them onto an agar plate, a petri dish, that has the antibiotic. So you would take these bacteria now and put them on a petri dish with ampicillin. Okay? And ampicillin will kill bacteria. They cannot survive in the presence of that antibiotic. But any bacteria that has taken in the plasmid, not only does that bacteria have the human insulin gene, it also has that other gene, remember, that we perhaps put on the other side of the plasmid, the ampicillin resistance gene. So this bacteria can survive in the presence of ampicillin. And so if you do it this way, you're going to know pretty quickly, if you were successful, if any of your bacteria survive and grow on that ampicillin plate. The only way they could have survived is if they did take in the plasmid. If they never took in the plasmid, ampicillin is going to kill them. But if they took in that ampicillin resistance gene, now they can survive in the presence of ampicillin. And we know they also have the human insulin gene because, remember, both of those genes are physically located on the same plasmid. So now that you know you've been successful, you allow this recombinant bacteria, this genetically modified bacteria, to do what bacteria do best. You give them nutrients, and you keep it warm, and you let it reproduce. And every 20 minutes, this bacteria cell is going to copy all of its DNA, including the genes that you have just introduced, and it's going to divide. Now you've got two bacteria that can make human insulin. And 20 minutes later, they're going to divide. And you'll have 4 bacteria that can make human insulin. 8, 16, 32, 64. It doubles every 20 minutes. So you can very quickly grow large quantities of these GMO bacteria, which will, all of them now will make human insulin. So at this point in the process, there's really a kind of three directions this can go. You can use these bacteria as they divide and reproduce to make many copies of whatever the gene is. Remember, I just used human insulin gene as my example, but it could be a lot of other genes. Whatever the gene is, I can use the bacteria now to make many copies of that gene. And then I can do research on that gene. I can introduce that gene into plants. If maybe it's a gene that will make my corn resistant to bugs, or maybe it's a gene I can put into soybean plants to make them more nutritious, or maybe it's a gene that I can introduce into other bacteria to alter them so that they can be used to clean up toxic waste. Or maybe instead of just using the bacteria as a copy machine for genes, maybe I just allow those bacteria to reproduce and make whatever the protein is, whatever protein that gene codes for. Maybe it's human insulin, maybe it's human growth hormone, whatever, whatever that particular gene of interest codes for. I can take that protein now, use the bacteria to make many copies of that protein, and then I can do research on that protein. I can use it to treat human disease, such as with the human insulin gene. Now, there's one big problem that I just glossed right over. I don't know if it occurred to you or not. But if you're really sharp and you've really been paying attention, you might have seen a problem with this. And that is the cell that I got the gene of interest out of is a eukaryotic cell. See the nucleus? If this is the human insulin gene, then this is a human cell. It's a eukaryotic cell. And so that gene, that little gene right there, has introns. And so if I put that gene into bacteria, the RNA polymerase in the bacteria can transcribe it, turn it into messenger RNA. That's no problem. But bacteria do not have spliceosomes. Bacteria don't need spliceosomes because their genes, bacterial genes, don't have introns. And so even if I put this gene into the bacteria, they're going to have no way of actually building a protein from it because they have no way of splicing out the introns. Oh, Lord, how do we solve that problem? Well, we solve that problem using a technique called cDNA synthesis, or complementary DNA synthesis. And it works like this. Okay, so here's how this works. Here is our gene of interest in a eukaryotic cell. Just to stick with that same example, let's say this is the human insulin gene. We're inside the human cell here. And notice this gene has exons, which we want to keep. It also has introns, which are going to need to be spliced out. So what you do, the way this really works, is you allow this gene to stay put. Leave it in the human cell for now. Allow it to be transcribed inside the human cell, just like normal. Both the exons and the introns are going to be transcribed. And then the normal RNA splicing process will take place. The spliceosome will come in and cut, get rid of that intron, cut, get rid of that intron. All the exons will be spliced together. So now we have this mature messenger RNA. No introns. And this is what you would actually extract from the human cell. Not the original DNA, but you would extract this messenger RNA after the normal splicing has taken place. Now you're going to take this messenger RNA, put it into a test tube with a very special enzyme called reverse transcriptase. Okay, now reverse transcriptase is an enzyme that's naturally occurring in some types of viruses. They're called retroviruses. That's not terribly important for you to know right now, but you're going to take this messenger RNA, put it into a test tube with reverse transcriptase, and what this wonderful enzyme does is it does transcription in reverse. So instead of going from DNA to RNA, which is what transcription is, it goes from RNA to DNA. So the enzyme reads the A's and the G's and the C's and the U's on the messenger RNA, and it creates a complementary DNA strand, just a single strand of DNA initially. So if there's an A on the RNA, it puts down a T here. If there's a U on the RNA, it puts down an A, G, C, C, G, you know, it's transcription in reverse. That makes a single strand DNA copy that has the same information as this edited messenger RNA. Then there's another enzyme that is added to the test tube to basically chop up this little piece of RNA and get rid of it. It's called degradation of the RNA. And then finally, a third enzyme is added into the test tube, which is just good old-fashioned DNA polymerase, so that we can take this single strand of DNA and go ahead and synthesize the complementary strand, because we know DNA is supposed to be double-stranded. So what you have, this finished product right here, is a double-stranded DNA molecule. This is the gene, the human gene, the human insulin gene, or whatever the gene of interest is. It's the same gene that we started with up here, but minus the introns. It no longer has introns. And so now we can take that cDNA version of the gene and insert that into the plasmid. And the bacteria will be able to transcribe it and translate it with no problem, because the introns have been taken out. Pretty cool. Now, there are two wonderful labs, DNA labs, that go along with this unit. I'd like to talk to you a little bit about one of them today. It's a colony transformation lab. And I'm just going to walk you through the basics of the setup, and then we're going to look at some hypothetical results, and then we'll be done with this lesson. So in this experiment, you are literally going to genetically modify some living E. coli cells. You're going to introduce a gene into the E. coli cells that will give them a phenotype that they did not previously have. Okay, so your basic setup is these sort of tan circles represent Petri dishes. Sometimes they're called agar plates because they have on their bottom surface a kind of jelly-like consistency of a nutrient material called agar. It's got protein and sugar, you know, it gives the bacteria their nourishment. So you're going to have 6 of these agar plates. 2 of them are luria broth plates, LB. That means they have nothing on them but nutrition. There's nothing deadly on the plates, just good old-fashioned bacteria food based on something called luria broth. The 2 of the plates will have luria broth, which is the good nutrient solution, but they'll also have kanamycin, which is an antibiotic that's very deadly to bacteria. And then 2 of your plates will have luria broth, which again is the nutrient, plus ampicillin, which is another very effective antibiotic that is deadly to the bacteria. Now you're going to have bacteria growing in test tubes that are labeled plus and minus. You'll see what plus and minus is all about in just a minute. Each test tube, you're going to introduce a small amount of cold calcium chloride, which is the liquid that will be suspending the cells in. You're going to put a small clump of E. coli bacteria into each test tube and mix that with the calcium chloride. So now you've got, I don't know, millions of bacteria floating around in this liquid. You're eventually going to add some luria broth, which is the nutrient solution that keeps the bacteria alive, keeps them happy. But notice one of the test tubes, only the plus tube, you're also going to introduce a very small amount of a liquid solution that contains plasmid DNA. So you're going to have, in this little tiny drop of liquid that you add to this test tube, you're going to have thousands of copies of little, let me back up and remind you, of these little circular plasmids. They're tiny, 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 microscopic. You can't see them, but they're there. And instead of a human insulin gene, your plasmid DNA is going to have one or two other genes, and I'll get to that in a minute. So only the bacteria in the plus tube have a chance to take in this plasmid DNA. There's no way the minus cells over here in this test tube can possibly be transformed or genetically changed because you're not introducing the plasmid DNA into the minus tube. It's just your control group, right? So the cool thing about this lab is it's a mystery that you have to solve because you don't know which plasmid DNA solution your group is going to be given. I'm just going to give you a test tube and say, here's your plasmid DNA, but you don't know which plasmid I'm giving you. Some of you will get the p-amp plasmid, which has the AMP-R gene. That's the gene for ampicillin resistance. Some of you will get the p-can plasmid, which looks like this. It has bacterial DNA, just like plasmids always do, plus an extra gene for kanamycin resistance. And then a third of you will get what's called the p-green plasmid, which is kind of a weird name. But that means it's bacterial DNA plus two extra genes, not one but two. So this plasmid has the AMP-R gene, which is the same AMP-R gene we had here, the gene for ampicillin resistance, plus a jellyfish gene called the GFP gene, green fluorescent protein. It's a gene that codes for a protein that sort of produces a green glowing effect in the cell. So this is pretty neat. Now, the basic idea is you will add all of these materials to your test tubes, as I explained before, and then you'll take a small puddle of the bacteria solution and spread them on each type of plate. And then you'll take a small puddle of the bacteria from the plus tube and spread them on each of these three plates. And really what you're looking for is what we see growing on those plates 24 hours later. We'll take all 6 of your plates and we'll put them in an incubator and we'll keep them at 37 degrees nice and warm for 24 hours, and we want to see what's growing on those plates. So I'm going to walk you through some hypothetical results. And so here's our 6 plates. And so one possibility is that you would get on both of your luria broth plates what's called a lawn. That means virtually all of the bacteria you spread on this plate and on this plate, those bacteria all survived. And they really should, right? Because these are luria broth plates. It doesn't matter whether the bacteria received the plasmid or not. They don't need antibiotic resistance genes to grow on a plate that doesn't have antibiotic. There's nothing on these plates to kill the bacteria. So they should flourish. They should survive. You should see a lawn on both of these plates. which is just like wall-to-wall bacterial growth, and it kind of looks milky as you see here. So here's a plate that has kanamycin, which kills bacteria. Notice, sure enough, I don't see any living bacteria on this plate. It looks just as clean as it did to start with. And then these two plates both have ampicillin, which is very deadly to bacteria, so of course none of the bacteria survive. But notice on this plate here, you see these dots? Each dot is a colony of bacteria that did survive. This is a kanamycin plate. Kanamycin kills bacteria, and yet we see evidence that some of the bacteria did survive on this plate. Each colony is made up of thousands and thousands of surviving cells, and right in the middle of each colony was one single bacteria that survived. One single bacteria that had the plasmid that contained the CAN-R gene that allowed it to be resistant to kanamycin. So from this result, which of these three plasmids did this group have? It's pretty easy to tell that the correct answer is this one. This group apparently was given the pecan plasmid, and that's why we see colony growth on the kanamycin plate. Now remember, the reason you don't see bacteria on this kanamycin plate is because, see that minus plasmid? These cells came out of the minus tube, and so they were never given the plasmid DNA. They had no chance to take in that CAN-R gene. Okay, different set of hypothetical results. We still have our lawn on both of the LB plates. That should always be the case. Notice no growth on either kanamycin plate, no growth on the ampicillin plate except for the AMP plus plate. Remember, the AMP plus plate would be the ampicillin plate onto which cells were spread from the plus tube. So they did have a chance to take in the plasmid. Which plasmid, which one of these three plasmids did these bacteria take in? Obviously, they took in the AMP-R gene if they're able to survive on an ampicillin plate. So, but that could be this plasmid or this one. So how do you know? Well, when I look at the color of these colonies, there's a lot of colonies on this plate, by the way. This group was very successful at transforming their cells. But the colonies are just sort of this normal milky white color that you would expect from E. coli. So there's no evidence of a green fluorescent protein being present in the cells. Let's look now. This is not quite as brilliant as I was hoping it would be. It just didn't show up well in the photograph. But do you notice a difference here? This time we have colonies of bacteria on an AMP plate, an ampicillin plate, but they have a greenish color instead of that normal milky white color. If you look at that same plate under ultraviolet light, it really glows in the dark beautifully. You can't really, it doesn't look all that impressive in normal white light. But this would be indicative of a group that received the pea green plasmid, because we see growth in the presence of ampicillin, meaning this gene is present in the cell. We also see a clear indication of GFP, that green fluorescent protein. All right, so I think that's all I wanted to cover. This is already 45 minutes long, and I do not like to go this long, so I apologize. This is just part one of our biotechnology lesson. Please keep reading pages 270 to 275 in your textbook, and that's in the Campbell Biology in Focus, second edition. And I'll be back soon for part two.